My name is Fowler. I'm a fifth-year graduate student in the lab of Daryl Bosco. I study a protein that is involved in about 97% of ALS cases, as well as a handful of other neurodegenerative diseases. It's called TDP-43, and it acts as a chaperone and a regulator of RNA, so the instructions for making proteins. And it's typically found in the nucleus, or the center of the cell, but in disease, it accumulates in the wrong parts of the cell, which impairs its ability to do its jobs, which are critical for cell health. So what we study is conditions where cells are stressed. So changes to their environment cause them to be stressed, which results in a process known as cellular stress response, which is important to the cells to be able to recover so they can handle the stress, recover, and get back to normal. TDP-43 in these conditions also changes its localization. So it goes from the center of the nucleus to other parts of the cell. And this is really not understood and also believed to be a really important event that may contribute to later on pathology that is seen with TDP-43 in these diseases. So my role and my focus is to understand what TDP-43 is doing during these conditions of cellular stress, as well as how that may play a role downstream when the disease becomes to progress. Not only is this question important because we'll understand the function of TDP-43, but it will provide insight into earlier on components of disease, either prior to or early in disease, which will allow us to identify biomarkers, which is really important, as I'm sure everyone knows, in the diagnosis of ALS, which right now is really hard to do. And for those who already have ALS and it's already progressing, we'll be able to identify novel therapeutics, which will hopefully be able to address a lot of the problems with the cellular health that we're seeing. In the models that we use, they're human-based models, which allow us to be closer to a translational and therapeutic opportunity sooner, but they're also expensive. They take a lot of maintenance, they require a lot, both in personnel and media and reagents and all sorts of things. So the funding is critical, and that's the best way for us to model the disease in the best way that we can and make more progress and advance the therapeutics